Hi, welcome back. In this video, we're going to be learning about blood typing and everything related to that. So when we're talking about blood typing, um, we're usually talking what's known as the ABO blood system. And that is like when people say, oh, I'm B positive or I'm O negative. What we are referring to are both phenotypes and genotypes. So a phenotype, just like in anything related to genetics, a phenotype with a P refers to physical characteristics. In this case, are you type A? Are you type B? Are you type O? Whereas a genotype is telling us exactly what are the genes involved. So I'm going to start first with the phenotypes and then show you how they relate to the genotypes. So in the ABO system, there's 4 possible blood types, A, B, AB, and O. And so I need you to envision a little red blood cell. And on the surface of the blood cell, there are proteins, okay? And in this case, the proteins we are looking for are the proteins related to the ABO blood system. So if you're type A, I want you to envision that on the surface are these little A molecules. Whereas if you're type B, you won't have A's, but you'll have little B's sitting on the surface. Now, if you're type AB, there is nothing called an AB. Type AB means you have both A's on the surface and you have B's on the surface. And type O, well, that never was an O. It used to be a 0, as in null, as in nothing. It means the surface is naked, okay? So no parent gave you any type of gene. So when you're looking at these pictures, I don't want you to think, oh, if you're A, it means you have little blue balls on the top. No, it just means you have this protein, which we have named the A protein. So what does that mean? So blood type A, well, everybody, you get 2 genes, one from mom and one from dad. So if you're blood type A, you could have gotten an A from each parent. And there's 2 ways to show this. The first way, the way that scientists, geneticists do it, is it's an I with 2 big A's on it. But this column here, this is not how most people in introductory science classes do it. You're going to do it like this column on the far right, where we're just sticking with the A's and the O's. So if you're blood type A and you got the A gene from both parents, then your genotype is AA. Whereas if you only got the A from one parent and the other parent, I guess, didn't love you because they didn't give you anything, well, no, what it meant was they just didn't give you anything. Then you could be AO, you could be heterozygous as opposed to homozygous where both genes are the same. Now, because O is nothing, A is going to be dominant over the O, and so both of these genotypes, AA and AO, both are blood type O's. By the way, if you're looking at this table, this first one, A with nothing, that correlates with the AO, and this next one correlates with the AA. I just realized that they are upside down from each other, the same thing for the B. So exactly how we talked about with the A, the same thing happens for the B. You could be homozygous BB, having gotten the B gene from both parents, or you could have gotten the B from one parent and nothing from the other parent. Now, the only way you can be AB is that you have one A and one B, which means one parent gave you the A. So that parent would have had to have been either AA or AO because they gave you an A. And the other parent gave you a B. So the other parent would either have to be BB or BO. That's how you got your B. And lastly, an O means you got nothing from either parent. I guess you're not their most favorite child. So how can you become OO? Well, if both of your parents are O, then obviously neither of them has anything to give you and you would be O. However, if you have a heterozygous A parent, a parent that is AO, they don't have to give you that A. They can give you the gene for nothing. And the same thing if you have a type B parent. They could give you the gene for nothing. So if you're two parents, one is an A, that's heterozygous AO, and the other is a B that's heterozygous BO, they could make you type O, having gotten nothing, and you could have a sibling that's type AB, having gotten an A from one and a B from the other. Or you could have a sibling that's type B that only got a B from one parent and an A from the other. Or you could have a sibling that's type A that got an A from one parent and nothing from the other. So this is how blood phenotypes and genotypes work. So understanding that, then we can figure out how this relates to getting blood transfusions. So and the way to do that is not to memorize it, but I'm going to have you make this table. Okay? So what I want you to do is just start making the table and add the columns as we go along. So let's start first with the far left column, which are the 4 blood types. So make one column, and then below it you have 4 rows we're going to make. So one for type A, one for type B, one for type AB, and one for type O. So I'll pause for a second while you actually write that on a piece of paper. Write it in your notes, wherever you are. Okay. The next column is, what is the surface antigen on the red blood cells in that blood type? So what's the surface antigen on an A? Well, you've got the pictures up here to help you. Try to do it without looking at the pictures. So, but what would you write here? What would you write in all 4? I'll give you a minute to fill them out. Okay, so it wasn't a minute. So obviously for blood type A, you would write A. For blood type B, you would write B. But for blood type AB, you need to make sure you write A and B. Don't be writing AB, because there is no surface protein known as an antigen. There is no antigen called AB. So you must write A and B. And then for blood type O, put a line through it, or write the word none. All right. Now that you know the antigens, what we're going to do next is look, what type of antibodies are you going to make? So what's interesting about blood is that your body will make antibodies against the other type of antigens. It doesn't do this for everything. Like for RH, it's not going to make it until it's exposed to it, but for blood, we're going to make some antibodies. So if you're blood type A, it means you have A's for the antigens. So what would you make antibodies against? What do you not have? That's right, it would be B. You don't have B. So you can't write the word B antibodies, because that's not what they're called. They're called anti-B antibodies. So A-N-T-I with a dash, then a B. Anti-B antibodies. And then if you get tired of writing the words antigen and antibodies, antigen is going to be abbreviated AG, and antibody is going to be abbreviated AB. So under antibodies made, if you're blood type A, you should have written anti-B antibody. So go ahead and fill out the other 3, and I will give you some time to do that. Okay. So what did you write for if you're type B and you have B antigens? Hopefully you wrote anti-A's. Okay. How about if you're type AB blood, the surface antigens were A and B? Would you be making anti-A's? No, because you have A's. Would you be making anti-B's? No, because you also have B's. So you wouldn't be making any antibodies. So make sure you write the word none. Okay. And then type O, you have no surface antigens. So you don't have antigens. Do you like A? So you'd be making anti-A's. And do you like B? No, so you'd be making anti-B's. All right. Once you have that column for antibody made, then you can figure out what types of blood you can receive. You can't receive a blood that contains the antigen that you make the antibody for. So I'm going to go through one row and then I'm going to have you pause it and you will do the rest on your own. And I'm not going to give you the answers to these. Okay, so type A has surface antigen A. You made anti-Bs. So can you receive A? Well, it's the same as you, of course. Can you receive B? Well, you're making anti-Bs. So no, you can't get B. Can you receive AB? Do you see the B in AB? Well, you're making anti-Bs, so you can't get blood that's AB. And can you receive O? Yeah, of course, because you're only making anti-Bs and O's have nothing. All right. Now, before you do check marks and X's for these boxes, you either need to write the word yes for one or no for the other, because as you go along and studying, you're not going to remember, does the X mean yes or does the X mean no? Okay. So go ahead and fill out, stop the video and fill out the rest of the table. Okay, welcome back. Hopefully you're able to work your way through that. I did not want you to rely on memory. I want you to realize that if you know the 4 blood types, you can figure out the antibodies, you can figure this out. So on a test or a quiz, there's no memorizing that needs doing. You can actually fill it out. And now look at what you have filled out and figure out where is the universal donor. Okay. The universal donor is going to be up here because this is receiving blood. These are the donor types. Which one of these 4 columns has yes? Has yes all the way down. That's the one that's a universal donor. Universal recipient is going to be looking here and looking across the rows here. Which one of these says yes across? I can receive A and B and AB and O. That's the universal recipient. So is the universal donor the same as the universal recipient? Only you will know once you have the table filled out. Okay. So looking in the US at blood types by race and ethnicity, what you will notice for all ethnicities, type O is the most common, followed fairly closely by type A. Type B is much less common, but you will notice in Asian and Black populations is significantly, like at least double the prevalence than in white populations. And that's why they are going to have higher populations of AB, because they have more of the B gene in their populations. So how about that positive or negative thing? That's another antigen, which is known as the D antigen. So we have ABOs and then we have a D. So the ABOs are giving us A, B, AB, and O. The positive negative comes from the D. And this is one where you're also getting genes from both parents, and getting the gene is dominant. So that if you get the gene for both parents, or if you get the gene from only one parent, that makes you Rh positive. So this plus plus, that would be DD. And plus minus would be D with nothing. I don't want to put O because it doesn't have anything to do with ABO. So that's why I went with pluses and minuses. Plus here meaning you got the D gene, and minus meaning this parent did not give you the D gene. So if you get the D gene from one parent or if you get it from both, that makes you Rh positive, which means the only way you're Rh negative is if neither parent gave you the gene. So the only way that's going to happen is if both parents are negative, then they have no choice, or if you have both parents being heterozygous. In other words, both parents only have one copy of the gene, then they could both give you the other copy of the gene that doesn't have the D antigen. So when we look at the incidence of this, it doesn't really matter about race or ethnicity. 80+% of all people are Rh positive, and a very small minority are Rh negative. And what you'll notice in the Asian community, it's even smaller. And that's because when we look worldwide, the Native Americans and Native Australians, 99% were Rh positive. Africa, 97%. East Asia, 95%. And if we look at Europeans, the lowest population in the world are the Basques, which is the southern part of France, closer to the west side. So it's kind of like the French-Spanish border. This is the population in the world that has the highest incidence of being Rh negative, which means it has the least amount of the G antigen. And even there, 2/3 of the population is Rh positive. And so since most white Americans are of European ancestry and a lot of Europeans are of French and Spanish ancestry, this is why the white population has slightly lower incidence of Rh positivity than the other ethnic groups. So why is this Rh important? Because not only do you have to have the same blood type so that you're not making antibodies against it, when you receive blood, if you're Rh negative, you're going to make antibodies against the G antigen. Now, these you're not going to make until after exposure. And so where it is important has to do with pregnant women. So if you look at this picture over here, this is the lady's breast area. So this is a lady and she is Rh negative. And she is making a baby with this man with a prominent prominent thyroid cartilage, the Adam's apple in his neck, and he is Rh positive. So they end up creating a baby. And lo and behold, this pregnant mother who's Rh negative, the baby is Rh positive. And so what happens, especially at the time of delivery, or at the time of a C-section, or at the time of a miscarriage, or the time of an abortion, when the baby comes out, it's a bloody process. Mom's blood vessels are torn, okay? And the baby's antigens can enter the mom's circulation. So the mom's circulation gets the Rh positive blood. And so the mom's circulation goes, hey, dude, we can't handle Rh positive. I'm Rh negative. And so I'm making antibodies against it. And so that's what all these little tiny white marshmallow things are. Okay? And so now this time I'm showing it's a second pregnancy. Okay. And so the mom has these anti-Rh antibodies, and these antibodies are small enough that they can travel in the mom's circulation, cross the placenta, and go to the baby, where they will attack the baby's Rh positive blood. Mom's anti-Rh or anti-D antibodies cross over and attack the baby's red blood cells, and the baby's red blood cells will then all die. Okay. And this disease has two different names. The first one's called erythroblastosis fetalis. This is not fatalis as in being fatal, as in killing. This is fetal as in a fetus, as in an unborn child. And the other name for it, which isn't in Latin, it's called the hemolytic disease of the newborn, because it used to be diagnosed at the time of birth, where basically because of the antibodies are attacking, all the baby's red blood cells are being attacked, and the baby ends up being profoundly anemic. And this actually was a lethal disease. Nowadays, at the first prenatal visit, mom gets her ABO blood type checked. If she's Rh negative, they can check for antibodies. You know, moms, it's super important to tell your your healthcare provider if you've ever been pregnant before, even if you had a miscarriage at 2 weeks, 4 weeks gestation is very important, because if you're Rh negative, they're going to follow your antibody levels. And then there is a medication whose name you don't need to know called RhoGAM, which actually prevents mom from making these antibodies that will attack the baby. So that's a really good thing. So what I would like you to do is read this slide about blood transfusions and get rid of the myths here in the United States. Everything is sterile. You're not going to get infected. They can't take too much. You are hooked up to a closed situation. And once it is full, it doesn't continue taking any more blood. None of you are too old. None of you are too young. If you're under 100 pounds, then you're too light. Your blood group is not too common. Your blood group is not too rare. We want everything. And there are very few medications that stop you from taking it. And if you are on a medication, it means you can go, that blocks you, it means in 3 weeks or 2 weeks or a month, you can go back and do it. So a lot of the blood is used for surgeries and for cancer treatments. Accidents. So really be thankful, because the people that need blood should not have to, as is in other countries, rely on family members and friends donating blood for them specifically, or else they die. Here in the United States, you can receive anybody's blood that's in the blood bank. Okay. Now, when we're receiving blood, we're not receiving the entire pint that you are donating. So that's all you're donating, a pint of blood. But it gets separated out. And so if you need the plasma or you need the clotting factors, that's what you get. If you need red blood cells, that's what you'll get. And this way, that 1 pint of blood can go to more than one person. All blood is tested. So it's super important in those screening questions you don't lie to them, because every pint of blood, they spend more than $200 testing it for about 50 different things. They'll tell you that, and if you come back, for instance, HIV positive, they are required to notify you. If you come back with hepatitis, they're required to notify you. If you come back with syphilis, they're required to notify you. Other viruses and disease markers are not required to notify you. Plus, the other benefit about donating blood is you get to find out what your blood type is, and then you'll know it for the rest of your life. And with that, we are done with this. I want to thank you so much, and I will see you again soon. Have a great day.